Hi everyone, my name is Adrian, and our research is focused on developing a nanoparticle formulation of a novel lipopeptide-based intranasal vaccine for SARS CoV2 infections. I would like to point out that this research kind of started back in around May, so lots of the research is still ongoing and thus fairly new. The preliminary studies started a while back in 2020, so all of the data we have is still coming as we speak. So, a little bit of background information or introduction. As we know, the SARS COVID-19 has infected over 770 million individuals globally. As we know, we've been masking up, keeping up to date with isolation protocols, and keeping up to date with our current vaccines. However, emerging variants continue to challenge these efforts. For instance, newer variants such as Omicron, Delta, and even subvariants of Omicron, such as BQ.5, continue to threaten the efficacy of our vaccines. And thus, this addresses the need for a cross-protective vaccine capable of combating these variant strains. So, recent advancements have shown that lipopeptides from SARS CoV2 spike protein induces pretty good protective B and T cell responses. And these lipopeptides aren't just derived from the spike protein. We also have the nucleocapsid protein and envelope protein as well. And so if we take a look at the schematic on the top right, I guess, there's the spike protein, and there's two different domains that we are aiming to target, the spike 1 and spike 2. And in this case, spike 1 refers to the RBD or ribosome binding domain, and spike 2 is the fusion protein. We also have a nucleocapsid and envelope protein, which I will talk more about at the end. And as well, we're going to be looking into a new kind of novel technology, which is the PLGA or polylactic co-glycolic acid, which is a type of nanocarrier. So nanocarriers in literature have been shown to improve vaccine stability, enhance epithelial penetration, and induce a more robust immune response. So on the bottom side of the diagram, there is an example of PLGA, which is what you can do with nanocarriers in general. You can add adjuvants, antigens, and even targeting molecules to elicit a specific immune response. Thus, we aim to address the global need for an intranasal coronavirus vaccine. And so our objective in this part of the research is to develop a nanoparticle formulation of a lipopeptide based from the LP1, so that's the spike 1, and LP2, that's spike 2, derived from the well-conserved regions of SARS CoV2 spike, aiming to develop a protective intranasal COVID-19 vaccine. So we hypothesized by incorporating these LP1s and LP2s into our PLGA nanocarriers, it has the potential to enhance immunogenicity from what we already saw in our preliminary studies, leading to a long-lasting, robust immune response and maybe a potential vaccine candidate. And so the schematic I have here kind of shows what the lipopeptide construct looks like. We have our lipopeptide, and we also have a lipid conjugate with the palomar foil tail, and the vaccine construct with the amino acids which is shown on the table. LP1 again is for spike 1, and LP2 is for spike 2. And so essentially this was a really big protocol, or we summed it down to our most recent protocol, but a lot of trial and error went into this because there's no protocol developed for our specific lipopeptides. So the first step is we would want to identify the nanoparticle loading optimization. So we would want to find good solvent for our lipopeptides. Then we decided to try varying PLGA concentrations at 5, 20, 25, and 50, just to check for the optimization of lipopeptide loading. And I want to point out that these numbers also aren't arbitrary. They have been shown in literature with the lowest kind of concentration that you can use and the highest concentration. So we decided to pull from those sources as well. And the negative controls were made for LP1 and LP2 without PLGA. Following, the nanoparticle characterization, the second step, which is the one that should go to, the size of aggregation were determined through zeta sizer, and finally lipid loading quantifications were determined using LCMS or lipid chromatography mass spectrometry. So some of our initial attempts when we first started gave us really big zeta size averages at around 6,200, which is way bigger than what we wanted for ours. And literature also shows that around 200 to 350 is ideal. And ethyl acetate for our lipopeptides were identified as LP1 and LP2, which was the ideal solvents. So moving on, we decided to characterize it with different concentrations, and this is where we decided to implement different concentrations. We found that at 50 milligrams per ml of the PLGA, the nanoparticles were still quite big, and the diagrams are actually on the other slide I'll show you in a second, but it's closer to the ideal size. So the table I have down below kind of shows you the what it looks like for the LP1 and LP2 without PLGA. So I do want to point out that lipopeptides tend to form in like micelles on their own when they're in solution. So we can actually kind of quantify their sizes as well. And we're given the size around 280 to 350. So starting with the first figure, when we decided to try and make these empty nanoparticles, we are getting kind of peaks at around still the thousands. However, the results for the quality report were good. And peak 3 is around 330, which is great. However, our intensity is around 9%, so that's still pretty low. Moving on for our size distribution, our nanoparticles at 50 milligrams per ml again, you can see that the volume, we're finally getting our results, which is around 300s, but volume is still at 2%, so there's lots of work that still needs to be done to kind of push it in that direction, and that's what our current studies are looking at right now. And so results for the LP1 peak, when we sent these off to get our lipopeptides commercialized and made, these kind of also came back with the NMR. So we decided when we wanted to look at the LP1 without loading, we can kind of identify where these peaks are. So as you could see, LP1, it came back with the spectrum at around 10,900, and LP2 similarly gave us 2 peaks, which correspond to our NMR. So what we would see if we had the controls ready, and if we were to do it with lipopeptides and PLGA, we would see that these peaks would kind of decrease over time so that we can see that encapsulation is actually happening, and then we can further quantify that at the end. Thus, as our protocol still has pretty high zeta average sizes, further modifications to the protocol are still underway. Noting efficiency is also just been sent off, so that is also results for the future. And other research that we plan to continue with is, right now we're at the step where we are creating our dose, but as soon as we're done, we plan to immunize our mice and also try to compare it with the market vaccines. And we also eventually want to incorporate the M&M proteins. Right now we have started on, and is still going on, we have obtained Pfizer vaccines, and we've decided to compare it with our lipopeptide and Pfizer. So eventually we decide to incorporate our lipopeptide and nanocarriers as well, and hopefully our M&M proteins as well. So I'd like to acknowledge our lab group and Dr. Afsani Lavasani for his lab group for helping us out with the PLGA, and Dr. Davies Neal's lab for LCMS. Thank you, and if you have any questions, you can feel free to ask.